All right, so welcome to video 3. In this, we're going to look at the neuromuscular junction and how contraction happens. All right, we call that the sliding filament model or theory. So, the contraction of skeletal muscle fiber requires the interaction of several chemical and cellular components. It sounds all fancy. Basically, nerves are going to go in and send a signal into this, and there's going to be chemical balances that are going to change and ions that are going to move and cause this muscle to contract. The results from a movement within the myofibrils in which the actin and myosin filaments slide against each other or past each other, shortening the sarcomeres. This is called the sliding filament model. And muscle fibers shorten and pull on the attachment point. So basically it's saying I've got a bone over here, and then I've got a bone over here, and I've got a muscle in between, and these little individual parts are going to pull together, and it's going to end up causing the one muscle to move towards the other muscle. Now, the way that it works is through nervous system contraction. So a neuron is going to send a signal to the muscle. Where this ends, all right, anywhere a neuron sends its signal, so the axon sends its little signal in through here to whatever else is around it. This is called a synapse. This particular area is called a synapse. Now, in skeletal muscle, it is called a neuromuscular junction. It is a special type of synapse. All right. Now, where this happens, this neuron that's bringing it in is called a motor neuron. The skeletal muscle fiber here is going to be underneath it. It's going to interact with it. The neuron is going to release something called a neurotransmitter, which is a chemical that's going to cause a reaction in the sarcolemma. All right, so the motor neuron, all right, is a neuron that controls skeletal muscle fiber. Motor neuron controls motor neuron is this, all right, so I've got my brain right here. I've got things that are bringing information in. Those type of neurons are called sensory, and then I have the brain sending signals out, and those are called motor. They can go to a gland or whatever, but in this instance, we're looking at when they go to a skeletal muscle. Where this part of the muscle is underneath this neuromuscular junction, this is called the motor end plate. It is going to have a lot of receptors that are designed to bind to the neurotransmitter. The space between the synaptic bulb and the motor end plate is called the synaptic cleft. There is a little, very, 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 very small opening between the neuron and the muscle. All right, and it's called the synaptic cleft, and this is where the neurotransmitter gets released. Inside the synaptic bulb, again, we are going to have little vesicles that are containing the neurotransmitters, and we call those synaptic vesicles. And these neurotransmitters are just simply the neurotransmitters, by definition, are chemicals released by a motor neuron to deliver a message to the muscle fiber. It really is more along the lines of the chemical that an axon releases to transmit the message to the next cell, whether it is a neuron or a muscle fiber or a gland or whatever. Neurotransmitters are how neurons speak to each other. So this is a picture of it. So you can kind of see this is the little synaptic cleft. This is a motor neuron. Inside of here, we've got synaptic vesicles. They are holding the neurotransmitter. This little folded in area is the motor end plate. These little dots are the neurotransmitters. Now, in skeletal muscle, neurotransmitters, the only neurotransmitter that is used with skeletal muscle is acetylcholine. Now, in the next few chapters, we'll get into the nervous system, and we're going to see that there are other types of neurotransmitters. This is by far the most common neurotransmitter. It is not, and it is the only neurotransmitter that skeletal muscle uses. All right. So if you ever get in trouble and you can't think of you had a question about a neurotransmitter and you can't think of what it is, guess acetylcholine. I can't tell you it will be correct, but it will be close. You've got the odds are on your side, I should say. So nerve impulses cause the release of acetylcholine into the synaptic cleft from the synaptic vesicles. They will attach to acetylcholine receptors on the motor end plate. This is going to cause, it says, membrane permeability of sodium and potassium ions. Basically, we're going to look in the nervous system how an axon sends a signal called an action potential, and it is the exact same thing. All it's saying is when this acetylcholine binds to the receptors, there's going to be an opening of these ion channels. It allows sodium to come in and potassium to go out, and it causes a wave of electrical charge. When this happens, it's going to pass along the sarcolemma, go into the cell by way of the transverse tubules, and cause the release of calcium ions from the sarcoplasmic reticulum. All right. Now, when we look at this, all right, I think sometimes it's tough to explain how they word things, but anyway, excitation and contraction coupling. It's the connection between muscle fiber stimulation and the muscle fiber contraction, because there is going to be a little time between it sending the signal and the actual contraction. We'll see that when we look at what a muscle twitch actually is. Now, when the muscle is not being called to contract, as we said, calcium ions are stored in the sarcoplasmic reticulum. Troponin and tropomyosin are bound together, and they are covering the binding sites that are on actin. Now, once calcium is released from the sarcoplasmic reticulum, they get into the cytosol, the fluid inside the cell. They will bind to troponin, and troponin changes its shape. As it binds to it, it changes its shape. At that point, tropomyosin, which is held in place by the troponin molecule, changes in shape, is caused to be repositioned. It alters its position because of the change in shape of troponin. So troponin sees calcium, binds to it, and in doing so, basically, it lets go of the tropomyosin. This causes the tropomyosin to slide off the binding sites, and now the binding sites on the actin are exposed. The myosin heads, which love them some binding sites, are going to bind to it, therefore binding to actin and causing what we call a cross bridge. In this picture, you see the cycle. So this is the way it looks when we have relaxation. Now, one thing that hasn't been mentioned up to now is the myosin heads are going to be holding something called ADP and phosphate. Now, if I was to be able to put this phosphate onto ADP, because ADP means diphosphate, I would change it to a molecule called ATP, which we're going to see here in just a minute. Now, when we look at how this works, this is, like I said, in relaxation. Right now, calcium has been released. Calcium is going to bind now to troponin. Troponin is going to cause tropomyosin to fall off of the little pimentos in this little string of olives that we looked at in the last video, but those little sites are called the binding sites. When the myosin heads see the binding sites open, they bind with them, forming cross bridges. Now, just like this is like a dance of greedy kids. So troponin is holding on to tropomyosin. It sees calcium, it grabs the calcium, lets go of tropomyosin, causes that to open up the binding sites. When that happens, the myosin heads bind to the binding sites, and in doing so, they have to let go of ADP and phosphate. Now, this is the key to the actual movement, because when they let go of the ADP and phosphate, we see that the myosin heads gets pulled forward. And since they're bound to actin, that means all of actin gets pulled forward, which means this little Z line gets closer to the middle. Now, when this ADP and phosphate get released, they get converted to ATP. And it turns out that ATP is the thing that these myosin heads love the most. Once ATP is formed, the myosin heads bind to ATP. They let go of the binding site. And when that happens, ATP breaks down into ADP and phosphate, causing the myosin heads to go back to their original shape. Now, as long as calcium is still bound to tropamine, that means the myosin heads, the actin site is still open for the myosin heads. So this cycle just repeats over and over again. Now, this is going to happen multiple, and when I say multiple, I'm probably saying hundreds of times in the course of a second. And this is like, if you can imagine a little caterpillar laying on its back and you put like a little a blade of grass on top of it, and all those little legs start grabbing it and pulling that grass forward. That's kind of what's happening here. And as long as those binding sites are open, it's going to continue to happen. Now, in the next video, we're going to look at a little bit more of this, and we're going to see how relaxation happens. But I hope you understand. Now, again, I can't stress enough, like I ended the last video, the most important thing is to understand what is where. And once you understand what is where, then like a bunch of dominoes, if you just understand the basic of what's where, you'll see how one little flick of a domino, letting go of the calcium, is going to cause a chain of reactions that's going to cause a muscle to contract. So I hope that made sense. I hope that this all works out for you. And again, continue to study this, and we'll see you in the next video.